Namaste students, Hamisha Khushro Bitia. I am JV'n's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth, Women's University, Jaipur. Today we are going to discuss the topic chromatographic separation process. And this topic comes under the subject upstream and downstream process. So let's start. So the purification of byproducts or the biological products by a bioseparation process typically involves a long sequence of steps, and each step requires the use of one or more unit operations. So the unit operations involves the filtration, extraction, chromatography and drying. Okay, so the basic principles and theory of the two key unit operations, filtration and chromatography, you have, and in this lecture we will focus on the chromatography. Okay, so what is chromatography? We have already have a brief description about the chromatography. So chromatography is a separation process. Okay, in this process, we will separate the molecules, the biomolecules, on the basis of the chrome. Chrome means color. Okay, so chromatography, the separation of mixture. It is used for the separation of the compounds in a mixture, or specifically the biological compounds. So in this, we passing a mixture dissolved in a mobile phase. Okay, so the mixture of the biomolecules is present in the mobile phase and through a stationary phase. Okay, we have a stationary phase through which we will pass the mobile phase, which separates the analyte to be measured from other molecules in the mixture and allows it to be isolated. So this process will allow us to isolate different molecules in the different fractions. So it uses the charge stationary phase and to separate the charged compound. Okay, so we can attach a charged molecule with the opposite charge if it is immobilized in the stationary phase. So resin that carries charged functional groups which interact with oppositely charged groups of the compound to be retained. Okay, so I explained that the opposite charge compounds will be retained by the stationary phase. So example is the FPLC. FPLC में. So what is ion? Ion is an atom or molecule which has lost or gained one or more valence electrons. Okay, if the electrons it have lost, then it will be positively charged, and if the electrons it gives it takes or it retains the more electrons, then it will be negatively charged. So anions are termed for the charged molecules which have the negative charge. Okay, so if a compound or a molecule have the negatively charged ions, then we will term it the anion, formed when an atom gains electrons. Okay, when an atom or molecules in a reaction gains the electrons, it will form the negatively charged ions, and which will be termed as the anions. So anions are negatively charged because there are more electrons associated with them, and then there are protons in their nuclei. Okay, so if one anion have protons in its nuclei and the electrons in its uh, outer the nuclei. Likewise, the cations are positively charged ions, and they form when an atom loses electrons in a reaction, forming an electron hole. Okay, if a molecule or atom loses the electrons, then the atom will be converted into the positively charged ion, and this will be termed as the cation. Okay, so the cation and anions have important role in the ion exchange chromatography. So what is the ion exchange chromatography? Simply the opposite charge ions will be captured by the stationary phase which have the complementary charges. That will normally ion exchange chromatography. Okay, so one another chromatography is there, that is the affinity chromatography. So affinity chromatography separates the proteins of interest on the basis of reversible interactions between it and its antibody coupled to the chromatography bead. Okay, here labeled the antigen. What it is in the affinity chromatography, the proteins are captured by their antibodies. Okay, so antigen antibody interaction will be there. Generally it is used in the pharmacological processes where the antibodies are produced, antigens are produced in the animals, and the pure form of antigens and antibodies are provided to the research purpose or the for the clinical purposes. Okay, so what is that in the stationary phase, either if you want to isolate the antibodies, then you will adhere the complementary antigens to the stationary phase, and if you want to isolate the antigens, then we coat or we attach the antibodies on the complementary antibody on the stationary phase, and the solution which have means the mobile phase which have the complementary molecules will be flow from the mobile phase. Okay, in the mobile phase, and it will be passed out from the stationary phase. Okay, so while flowing with the mobile phase, the opposite antigens or antibodies will be captured in the stationary phases, and after attaching there, that will be afterward eluted out with the with change in the pH or in the temperature, etc. Okay, so the pH will be changed, and uh, after changing the pH, the interaction which is formed between the antigen and antibody will be break, and we will collect the antibodies or the antigens. So with high selectivity, high resolution, and high capacity of protein of interest, purification levels in the order of several thousand fold are achievable. Okay, because antigen and antibody are the highly selective molecules, so purity will be improved. Okay, so high resolution, high capacity, high yield will be there, and purification will be there. So protein of interest is collected in a purified concentrated form. Biological interactions between the antigen and antibody, the protein of interest. Okay, so it can result from electrostatic interaction, van der Waals forces, hydrogen bonding. Okay, so the antigen antibody interaction can be created by these type of the interactions. Okay, so they may have the electrostatic interaction, van der Waals interaction, or the hydrogen bonding. Okay, so to elute the protein of interest from the affinity beads, the interaction can be reversed by changing the pH or ionic strength. Okay, so whatever the molecules are interacted or attached will be eluted out at the final step by changing the pH or the ionic strength. Okay, so to concentrating effect, the concentrating effect enables large volume of to be processed. The protein of interest can be purified from high levels of contaminating substances. Okay, so we can use the large volumes also here. The setup will be enhanced, and to making antibodies to protein of interest is expensive. So affinity chromatography is the least economic choice of the protein production chromatography. Okay, because when the antibodies are produced, how we produce an antibody? We insert the antigens in animals, and the immune system of animal um, generate the antibodies against the inserted antigen. So the antibodies production is a uh, expensive process. So we want to capture or collect all the antibody molecules which are produced in an animal. So affinity chromatography is a inexpensive or economic uh, choice to isolate the antibodies. Okay, so this is the pictorial representation of the affinity chromatography. Here the antigens and antibody are present, and these beads have the antibodies, okay, or antigens, whatever the opposite charge. So here the beads have the antigens, and with the flow in the mobile phase, the antibodies are there, okay, the red color antibodies are there, and when they pass out from this column, they will attach with the antigens, okay. And then after finishing the process, these attached antibodies will be eluted out or be collected by changing the pH. So another chromatography is the size exclusion chromatography. So it is a basic or simple form of the chromatography, where it is also known as the gel permeation or filtration chromatography. Okay, it separates the molecules according to their size. His name says size exclusion. So basis is the size. So low resolution polishing and tertiary quaternary structure netting. So what is that? That here we use some gel type of the molecules. So gel is there, and the gel have the various uh, pore size. Okay, so the pore size can be increased or decreased by changing the concentration of the gel. Okay, so if the pore size is greater, then the larger molecules will pass out fast. If the pore size is lesser, then the smaller molecules will be filtered out first. So on the basis of your choice that you want to elute first larger molecules or the small molecules, we will um, design the create the pore size of the gel in the gel permeation chromatography. Okay, so another is chromatography technique is a liquid chromatography technique. So here the mobile phase is liquid and it carried out either in a column or a plane. So here it is the column. Okay, this column has have the mobile phase. Okay, so the mobile phase will be passed out from this column. So in the HPLC technique, the sample is forced to a column that is packed with the irregularly or spherically shaped particles or porous monolithic layer stationary phase by a liquid or the high pressure. So what is that in the HPLC, high pressure liquid chromatography, HPLC is a type of the liquid chromatography where we use a high pressure specific pump which will force the mobile phase to move. Okay, so here in the column, stationary phase is already there. Okay, and in the mobile phase where our compounds are there, so that compounds will be captured in the stationary phase, and that will be eluted out afterward. Okay, so that is the liquid chromatography. So thank you. Hope you are satisfied with this digital session. And if you have any query, please write in the comment box. And if this session is powered by digital version 2, Jyoti Vidhyapeeth Women's University, Jaipur. Thank you.